Okay, good morning. So, the pharmacology of the autonomic nervous system is one area where students tend to have a lot of trouble. And the reason is I call it mental gymnastics. There are so many mental gymnastics that take place that just memorizing everything is going to be next to impossible. It really takes understanding it. So it takes getting into the details of things and then kind of understanding things. And so when we talk about the autonomics or the autonomic nervous system, we have two parts. We have the sympathetic nervous system and we have the parasympathetic nervous system. But recognize that the autonomic nervous system is a part of our peripheral nervous system, not necessarily our central nervous system. And the autonomic is just that. It's automatic. Okay. What we tend to find is that the sympathetic nervous system and parasympathetic are mostly opposites of each other, where this one will, you know, turn up heart rate and contractility. This one will turn down heart rate and contractility. And then also for the purposes of this part, we'll get rid of or we'll not talk about the enteric nervous system as much. So our focus will be on sympathetic and parasympathetic. And it's controlling mainly three different things. Our autonomic nervous system controls heart rate and contractility. It controls smooth muscle contraction as well as relaxation. And it controls the secretion of various juices in the body. Those are the main things. Remember, smooth muscle is everywhere. It wraps around the bronchi in our lungs. It wraps around our vascular system. It's part of the GI tract, which causes peristalsis or whatever. And smooth muscle can only do one of two things. It can either contract or relax. That's all it can do. It can't do anything else than that. And so what we have here is a map that I drew of the autonomic nervous system. And, everything in our body starts in the central nervous system. But what we have here are a couple of different things going on. We have our sympathetic nervous system arc, we have our parasympathetic nervous system arc, and we have our somatic motor neuron system, which causes voluntary muscle contraction, skeletal muscle, being able to walk to class or whatever. Okay. We'll remind ourselves that the sympathetic nervous system is responsible for our fight or flight response, and the parasympathetic nervous system is responsible for the rest and digest. Okay. What we find with, and we'll start at the top here, what we find with our somatic motor neuron system is we have a single myelinated fiber that carries signals from the central nervous system to the skeletal muscle to produce contraction using a neurotransmitter acetylcholine. In the autonomics, we have usually two neurons in series, which synapse at some sort of ganglia. And the ganglia are in different places depending on what system we're talking about. What we find is only the preganglionic fibers are myelinated, and acetylcholine is the neurotransmitter found in all postganglionic parasympathetic fibers, and norepinephrine is the neurotransmitter found in postganglionic sympathetic fibers. With the sympathetic nervous system, we find that fibers exit the central nervous system in the thoracic and lumbar regions of the spinal cord. Okay. And most sympathetic ganglia are found in chains on each side of the spinal cord called the paravertebral ganglia. Postganglionic fibers are long and multiple, so they can have effects that can be very diffuse and widespread, like increasing heart rate and telling the adrenal glands to release epinephrine and telling the blood vessels to cause vasoconstriction all at the same time. And that's kind of nice because the sympathetic nervous system can be activated all at once, fight or flight. In the parasympathetic nervous system, preganglionic fibers exit the central nervous system via the third or seventh, ninth, and 10th cranial nerves, so that oculomotor, the facial, the glossopharyngeal, and the vagus cranial nerves. And it's usually seen as well as 2 through 4 sacral nerves. Parasympathetic ganglia are usually found in the tissue that it's innervating. So postganglionic fibers tend to be very short, and the effects are more localized as you would compare to the sympathetic nervous system. Again, in many cases, the sympathetic and parasympathetic systems have opposite effects. However, parasympathetic tends to be more localized because of the close ratio of the ganglia, and the sympathetic prepares us for fight or flight, so it tends to be more diffuse with its ganglia located in the paravertebral ganglion. Obviously, there's going to be some exceptions, but one thing I want you to notice right now is whether we're talking about the skeletal muscle, whether we're talking about the paravertebral ganglion of the sympathetic nervous system, or we're talking about the ganglia of the parasympathetic or right at the tissue, they all use acetylcholine. Okay? Now, they'll use different receptors, but they all use acetylcholine. The only time we use a different neurotransmitter is at the tissue of the sympathetic nervous system, where we use norepinephrine instead of acetylcholine. And one thing I kind of want you to imagine in your mind is, all right, let's say we increase the levels of acetylcholine in somebody's body by 100 times. What effect would you see? Well, you would see that it would probably cause skeletal muscle contraction. It'll cause increase of the sympathetic nervous system, and it'll cause increase of the parasympathetic nervous system. But the major effect that we would see is parasympathetic overload. And that's because if we increased acetylcholine by 100 times, then we're going to increase it here and we're going to increase it here. So we're getting a 2 times effect on the parasympathetic versus a 1 times effect on the sympathetic, as well as on skeletal muscle. Obviously, there's going to be some exceptions to this model that I drew. And some of the major exceptions are on the adrenal medulla and the sweat glands. The adrenal medulla itself can be seen as a special sympathetic nervous system ganglion, which is innervated by preganglionic sympathetic fibers that actually release acetylcholine, not norepinephrine. Activation of the adrenal medulla forces the release of epinephrine from chromaffin cells into the bloodstream. And this is kind of a good time to differentiate that norepinephrine is a neurotransmitter and epinephrine is a hormone. Okay, so we never see epinephrine being used as a neurotransmitter, only norepinephrine. The next place is sweat glands, which are involved in temperature control. And at these places, postganglionic sympathetic fibers actually release acetylcholine, not norepinephrine as we usually see. These will activate eccrine sweat glands for that temperature control. This is a relative slide that I have made just to kind of get our brains accustomed to what we would expect finding. One thing I want you to think about going forward is what the role of the sympathetic and the parasympathetic nervous system is. Okay, so let's say we know the sympathetic is fight or flight. So in fight or flight, I would suspect or assume that I'm going to have elevated blood pressure. My eyes are going to dilate because I need to be able to see at night to run away from the tiger. My lungs are going to dilate so I can get as much air in as possible. I'm not going to want to poop or pee running away from a tiger. So my gut is going to turn down. My bladder is going to turn off because I won't want to poop or pee while I'm running away from a tiger. My sweat glands are going to turn on so I can regulate my temperature control as I'm running away from the tiger. My heart rate is going to increase because I'm really afraid. My liver is going to cause production of glucose so that while I'm running away, I can have a steady supply of energy. And those are the kinds of things I want you to think about. And so then we would expect the parasympathetic to do the opposite. We would expect the parasympathetic to cause pinpoint pupils, or decrease in heart rate, or bronchoconstriction, and or increase gut motility. And we'll find that our parasympathetic nervous system wants us to make secretions and digestion juices and eat nutrients. And it's going to make us poop, and it's going to make us pee, and things like that. And those are the general thoughts that I want you to have going into your mind. Is the thing you can always go back to is, okay, what would I expect to happen at this place? And based on that expectation, you'll be able to guess which receptors you would consider finding in those spots. And you'll find that most of the time when you do it that way, you'll be right. So let's go back and get a closer look at what's going on in the various sites of, let's start with the parasympathetic system, but let's see what's going on at the various sites of both the parasympathetic and sympathetic nervous system. Again, acetylcholine is released at almost every synapse in both systems except the location of the tissue, where sympathetic nervous system releases norepinephrine as the neurotransmitter. Okay? We're going to focus on each system and the effects they have on neurotransmission on that postganglionic synapse. And neurotransmission obviously requires 5 steps in order to have an action and function. You have to synthesize the neurotransmitter, you have to store it, you have to release it, you have to have that that transmitter have an effect on a receptor, and then it can't stay around forever. You have to inactivate the neurotransmitter. Again, norepinephrine is only found at the tissue, but upstream at the paravertebral ganglion, there is acetylcholine. So these are the places we'll be focusing on first, whether at the parasympathetic right at the tissue or the sympathetic right at the tissue. And the first one we'll talk about actually is the cholinergic or muscarinic transmission sites, okay? When I say the term cholinergic receptors or muscarinic receptors, they are the exact same thing, okay? So over here on our left is our parasympathetic postganglionic fiber. We just zoomed in on it here, which uses acetylcholine. The neuron on the left is the postganglionic neuron, and the right is the tissue that it's having an effect on, like the heart, for example. Acetylcholine is made by combining choline, which is a quaternary ammonium alcohol, to acetyl-CoA, which esterifies the choline. And the quaternary component is important because acetylcholine is stored in vesicles, and you don't want a lipid-soluble neurotransmitter stored in a lipid vesicle, or it would just leak out and you would have no control over it. And so this is when it's stored in vesicles in the nerve terminal. And what promotes the release of acetylcholine? Calcium. So what happens is an arrival of the action potential depolarizes the membrane, which opens voltage-dependent calcium channels. That influx of calcium triggers exocytosis of acetylcholine-containing vesicles that fuse with that cell membrane and release their contents. The empty vesicles will eventually then be recycled, but that neurotransmitter, that acetylcholine, can then have an effect of binding the receptor on the target tissue. In this case, it's the muscarinic M3 receptor or the cholinergic M3 receptor, which actually is one of the more important parasympathetic receptors in our body. Postsynaptic muscarinic receptors are also G protein linked receptors, which will lead to elevation of calcium and the effect on the tissue that you're looking for. We also have M2 receptors on the presynaptic neuron, and activation of those M2 receptors on the nerve terminal can inhibit the further release of acetylcholine. Essentially, it's causing feedback inhibition so that you don't release too much or you don't release too little acetylcholine during membrane depolarizations. Once acetylcholine has its effect, it can't just stay around forever. Remember, we esterified choline with acetyl-CoA, so we can take advantage of that by using an esterase system or an esterase enzyme like acetylcholine esterase, which hydrolyzes acetylcholine and is found in the membrane of the postsynaptic cell, which breaks it down into acetate and choline. And about 50% of these broken down products will eventually re-enter the nerve terminal, be resynthesized and recycled. Any acetylcholine that escapes out from the synaptic cleft and can make it out into the bloodstream or whatever, usually is going to be rapidly inactivated by butyrylcholinesterase, also known as plasma cholinesterase or pseudocholinesterase, in the bloodstream. So it can't have a huge effect outside of the local area that it's trying to be at. There are drugs that can interfere with this mechanism of neurotransmission, and one that we use all the time are going to be drugs like cholinesterase inhibitors, which will block the breakdown of acetylcholine and therefore leave elevated amounts of acetylcholine in the synaptic cleft to promote and enhance the effects of acetylcholine. Another thing is botulinum toxin, which will actually works to block the fusion of those vesicles with the membrane and hence the release of acetylcholine. So botulinum toxin can decrease acetylcholine release. And we also have drugs that can be both muscarinic receptor agonists and antagonists, which are important to act on that target tissue. So they're either muscarinic receptor agonists or antagonists. Another name for them is cholinergic medications or anticholinergic medications, because muscarinic and cholinergic are synonymous with each other. Now let's look at adrenergic neurotransmission, such as seen in postganglionic sites of the sympathetic nervous system. And you hear the term I used, adrenergic, and that's because in the United States, we usually say epinephrine and norepinephrine, versus in the UK, they say adrenaline and noradrenaline. So epinephrine is the same thing as adrenaline, and norepinephrine is the same thing as noradrenaline. And so we use the terms adrenergic receptors synonymously with our sympathetic nervous system. All right, well, so what about sympathetic adrenergic neurotransmission? And it's a little bit more complicated in the synthesis, at least, of its neurotransmitter. Tyrosine, the starting compound that we start with is called tyrosine, which is an amino acid found in proteins. And the first process is that tyrosine gets hydroxylated by tyrosine hydroxylase, which is the rate-limiting step in the synthesis of our monoamines. If you don't know, our monoamine neurotransmitters are things like serotonin, dopamine, and norepinephrine. But now here we have a product with two OH groups on the benzene ring, known as dihydroxyphenylalanine, or DOPA. And DOPA is then acted on by a carboxylase, which removes the carboxyl group, and this compound is no longer an amino acid now. Now it's just an amine, which we call dopamine. And dopamine is brought into nerve terminals and vesicles via the vesicular monoamine transporter type 2, VMAT2, and then it's hydroxylated quickly into norepinephrine, where it's stored in the nerve terminal. And if we are in the chromaffin cells in the adrenal medulla, the addition of a methyl group is added, which gives us the hormone epinephrine. And it's important to recognize that both of these are what are known as catecholamines because of the catechol group, or with those two OH groups, essentially, okay? Anything with two OH groups are catechols. So technically, dopamine and DOPA are catecholamines as well. But these are the very potent versions of those, and it's where that term comes from. In that postganglionic adrenergic junction, dopamine is synthesized and brought into vesicles by VMAT2. Dopamine, again, is quickly hydroxylated into norepinephrine, and an action potential activates voltage-gated calcium channels to force exocytosis of the vesicles containing norepinephrine, similar to everything we've seen before. Empty vesicles we know are going to be recycled, but the target tissues will either have an alpha or beta receptor, which will have, after bound to norepinephrine, have an effect on the target tissue. Adrenergic receptors come in a lot of flavors. Some examples are alpha 1, alpha 2, beta 1, beta 2, beta 3, and they're all G protein linked receptors. What we'll find in the next lecture is that norepinephrine and epinephrine are very different in the sense that we find epinephrine binds to all adrenergic receptors that we know of, whereas norepinephrine binds to all of them except for beta 2 receptors. And then because dopamine and dopa and those things were precursors, anytime in biology there's similarity in structure, means that there can be cross-reactivity and similarity in function. What we'll find is dopamine not only binds to dopamine receptors, but can also bind to beta 1 and alpha 1 adrenergic receptors. But again, we'll learn that more in the future. Activation of alpha 2 receptors on the nerve terminal inhibits excessive release of norepinephrine, similar feedback inhibition that we saw before. But again, norepinephrine cannot just stay around forever, and roughly 5% of it is uptook into the tissue itself by the OCT3 enzyme, similar to the OCT1 that we saw with metformin. But probably the most important mechanism is reuptake of norepinephrine into the nerve terminal via a transporter called norepinephrine transporter. This transporter also transports dopamine and tyramine, because those are also monoamines. And norepinephrine transmitter is found on a lot of different presynaptic membranes and cell types, like in the central nervous system, or for things like that. Anyway, I'm going to get too ahead of myself. NET can also reuptake other monoamines again, but once norepinephrine is reuptook, it could either be broken down by an enzyme known as monoamine oxidase to be recycled, or it can be repackaged and used again, which is what happens to around 70% of what's taken up, which is most of it. If some norepinephrine is able to slip by that norepinephrine transporter and escape out into the bloodstream, two enzymes exist in the blood in various organs. These are known as catechol O-methyltransferase and monoamine oxidase out in the system, which will also break down norepinephrine into inactive metabolites. You can see here that catechol O-methyltransferase and monoamine oxidase can exist in different areas. COMT is found in the liver, the kidneys, and other places, and monoamine oxidase is found in the nerve terminals, gut, liver, also all over the place. And those two enzymes will convert catecholamines into different metabolites. For example, COMT will produce an intermediate metabolite known as normetanephrine, which can be further broken down into vanillomandelic acid by monoamine oxidase. And these are important because if you don't know, these metanephrines, or vanillomandelic acid, can be measured in urine. And in cases where the adrenal glands are producing too much epinephrine, these urinary products will be significantly elevated. So we tend to measure these urinary products in patients who have pheochromocytomas. There are ways to manipulate this system as well. Adrenergic receptor agonists and antagonists can activate these receptors without norepinephrine being present at all. Other things like cocaine and tricyclic antidepressants, for example, can inhibit the reuptake of norepinephrine, leaving a lot more norepinephrine in the synaptic cleft to have its effect. Amphetamines and tyramine, for example, can actually enter the nerve terminal and stimulate norepinephrine release without needing neural stimulation at all. And monoamine oxidase inhibitors, which are antidepressants, can inhibit the inactivation of norepinephrine. And so it can increase the amount of released upon neural stimulation. So just some ways that we can augment this system. Now let's look at neurotransmission that occurs in all the autonomic ganglia, as well as the neuromuscular junction, so like voluntary skeletal muscle. And we'll be looking at these sites now. Remember, we just first looked at this site, and then we just looked at this site. But now we're going to look at these three different sites, which all use acetylcholine as their primary neurotransmitter. And of these three sites, we'll first look at the autonomic ganglia first, and then we'll look at the neuromuscular junction. So the parasympathetic and the sympathetic nervous system. And this is where I want you to make a good distinction here. The second class, so the first class of acetylcholine receptor we talked about were the muscarinic receptors, also known as the cholinergic receptors. The second class of acetylcholine receptors are called nicotinic receptors, and they're very, very different from muscarinic receptors, okay? Obviously, we're going to have the same production and synthesis of acetylcholine, all of that packaging and release in calcium and exocytosis that we saw before. But when acetylcholine is released at these, it requires two acetylcholine molecules, which will bind both units of the nicotinic receptor. And nicotinic receptors are ligand-gated ion channels with 5 subunits. Okay, the nicotinic N, or Nigel, are found in neurons, including the autonomic ganglia, as well as that adrenal medulla in multiple isoforms. Two bound acetylcholines will activate and open this ion channel, which will allow the influx of sodium, which will trigger an action potential in the post-ganglionic neuron. Okay. What we'll find is drugs like nicotinic, I'm going to get ahead of myself here. Acetylcholine is the only physiologic agonist for these receptors. And termination of action is going to work exactly the same way too. We have acetylcholinesterase. We have butyrylcholinesterase to diminish these things. But one thing I want you to remember is that nicotinic receptors and muscarinic receptors are very different from each other. So although they both use acetylcholine, there is no cross-reactivity between muscarinic agonists, we'll say. Muscarinic agonists have no effect on nicotinic receptors, and we'll say nicotinic receptor agonists have no effect on muscarinic receptors, even though they use the same acetylcholine, okay? Then we'll look at the nicotinic mic, M muscle receptor, seen only at voluntary skeletal muscle. And nicely enough, you'll see that there's really no difference between the nicotinic M, as in mic, and the nicotinic N receptor. They're going to have the exact same effects and the exact same getting rid of acetylcholine, okay? But this is how we move muscle, voluntary skeletal muscle, okay? Nicotinic receptor agonists and antagonists can be used to modify this system. Probably the most well-known nicotinic N receptor agonist is nicotine, okay? Botulinum toxin can also bind to and decrease exocytosis of acetylcholine from vesicles. And we can still use those same cholinesterase inhibitors as we saw before when we were talking about muscarinic receptors. Now that we have an idea of the different neurotransmission, now we need to at least get an idea of what happens upon activation of the receptor. So those beta receptors, or those muscarinic receptors, or those nicotine, or whatever. But in this case, we're going to be focusing mainly on the parasympathetic and sympathetic nervous system, okay? And this is that same slide. So that same slide I talked to you about before. Beside all of these are the general receptors that are used. The parasympathetic nervous system's easy because All of them except the heart mainly use M3 receptors to have their effect. The difference becomes with the sympathetic nervous system, which is not as intuitive or straightforward. Alpha is the dominating contractor of smooth muscle, so like alpha 1. Beta 1 is only seen really in the heart to increase heart rate and contractility, which is what you would expect with sympathetic tone. And then another important one is beta 2, which we generally only find in the lungs to cause bronchodilation, as well as to promote gluconeogenesis by the liver, so you can run from the tiger, as well as to increase blood flow to muscles. But we'll talk about these a little bit more in depth. But this is a good slide to always come back to. And again, here are the different receptors that we have for things. Cholinergic receptors, like muscarinic 2 and 3 receptors, which are the most important. We have 5 of them, but the other ones here are more important in the central nervous system. We have our nicotinic N and M receptors. We have adrenergic receptors, which have 2 families, alpha and beta. Our alpha 1A and alpha 2 receptors are the most important. Regard to beta receptors, beta 1, 2, and 3 are important. And then we have some dopamine D1 and D2 receptors. And it'll be important to know which sites you'll find these receptors and what agonists and antagonists can bind to them and what effects you would expect to see. And before we talk about the drugs, we need to see these areas a little bit closer, okay? So let's see how the sympathetic nervous system and parasympathetic nervous system affect these 3 main things: heart, smooth muscle, and secretion, like I said before. These again are some summary slides that I want you to look at on where you'll find the most common areas of these different receptors, whether it's in the parasympathetic system or the sympathetic system, okay? Before we talk about these autonomic inputs on the heart, for example, we need to remember how the heart contracts and relaxes. So contraction, I've said a couple of times now that calcium is the main doer of the body, and this is no different. Contraction of the heart requires calcium. What happens is an action potential coming from the SA node is going to cause opening of voltage-gated calcium channels. And this positive charge inward triggers the opening of calcium channels on the sarcoplasmic reticulum to quickly increase the amounts of cytosolic calcium. Calcium is going to bind to troponin C, which relieves its inhibition of the interaction between actin and myosin, and the muscle contracts. But our heart can't just stay contracted. And could you imagine if your heart just contracted and then never relaxed? You would die within a matter of minutes, seconds. So relaxation is just as important to contraction. To relax, we need to quickly remove all of that cytosolic calcium. And we have a couple ways to do that. We have ATP-driven calcium pumps in the plasma cell membrane, which work to pump calcium out of the cell. Remember, almost every cell in our body has a sodium-potassium ATPase on the cell membrane, which works to keep sodium on the extracellular side and potassium on the intracellular side. And so this sodium-calcium exchanger is using that gradient to drive sodium inward down its concentration gradient in exchange for moving calcium outside of the cell. We also have those same ATP-driven calcium pumps on the membrane of the sarcoplasmic reticulum, which will pump calcium and fill up the sarcoplasmic reticulum with calcium to be ready for that next contraction. And once all those calcium levels leave and decrease, calcium will dissociate away from troponin C and inhibit the action between actin and myosin, causing relaxation. And so now that we understand contraction and relaxation on heart muscle, let's see what happens to the heart with input from the sympathetic nervous system on beta 1 receptors. And these membrane receptors are going to be linked to a G protein, which can be a couple of different variants, either G sub S, G sub Q, or G sub I for inhibitory. In this case, beta 1 receptors are G sub stimulatory or G sub S linked. And the process starts with the binding of epinephrine or norepinephrine to those beta 1 receptors, which causes a conformational change of the receptor and activate G sub S. Okay? G sub S is usually bound to GDP, if you don't remember, which is guanine diphosphate. And when you activate it, GDP is released, and GTP, guanine triphosphate, binds to the G protein. The activated G sub S, or stimulatory protein, goes to an effector protein like adenylate cyclase, which activates it. And adenylate cyclase is interesting because it's going to cut the GTP into GDP, which will turn the G sub S back off to be reused again. But adenylate cyclase, once it's activated, is going to work to convert ATP into another very, very important structure here known as cyclic adenosine monophosphate. So ATP is adenosine triphosphate, and adenylate cyclase is going to convert it into cyclic AMP. Okay? Cyclic AMP can do a couple of things. Its major thing is going to be activating another protein in the cytosol known as protein kinase A. And remember, anytime you hear the word kinase, it means that it essentially just phosphorylates things. And pKa can have a lot of different actions. pKa can phosphorylate those calcium channels, leading to an opening of those calcium channels and rushing in of calcium. In addition, that rushing in of calcium is going to trigger voltage-gated calcium channels on the sarcoplasmic reticulum to open, which will lead to an influx of cytosolic calcium. Okay? By phosphorylating phospholamban, this will increase energy-dependent reuptake of calcium into the sarcoplasmic reticulum. Thus, it's going to pack calcium in the SR like sardines, which will make the next muscle contraction even stronger. And pKa can also phosphorylate troponin C, which can inhibit the interaction of calcium to troponin C, allowing it to dissociate quicker. And by doing this, it allows the heart to contract but also relax quicker, which can allow for a quicker heart rate. Another interesting thing we find is that an elevation of cyclic AMP can also increase SA nodal cells to increase their funny channel current production. And if you don't know yet, the SA node can automatically create action potentials and does this by funny channel automaticity. We'll talk about this more in the cardiology section. And so the end result is that epi or norepi on beta 1 receptors is going to increase heart rate and contractility. But what about the parasympathetic nervous system, or muscarinic or cholinergic system? Well, activation of M2 receptors activates G sub inhibitory linked protein, which has no stimulatory properties whatsoever. Okay, and it goes and it binds to adenylate cyclase, and it does not activate it. Okay, actually it blocks the ability for G sub S proteins to bind onto adenylate cyclase. The effect is that cyclic AMP levels will be reduced, thus decreasing calcium channel activation, phospholamban's ability to increase calcium ATPase, the effect of troponin C, and the effect on the funny current. And therefore, what we'll find is the major effect of acetylcholine and muscarinic activation on the heart is causing relaxation and decreasing heart rate and contractility. Interestingly, as a side note, what we find is that in the atria, potassium channels also open, which increases the polarization of the cell, which in turn will decrease the opening probability of calcium channels and even further slow calcium influx. But that effect on ventricles is much smaller than on the atrium. And we really only see this if contractility is already increased by the sympathetic nervous system activity, i.e. cyclic AMP was elevated. But neither here nor there. It's just a interesting extra note, if you will. Let's keep going here. And so that's how the heart kind of works in regard to our autonomics. But we have that figured out. What about smooth muscle? And where is smooth muscle located again? Boy, everywhere. Our blood vessels, our gut to do peristalsis, the bronchioles and bronchi to bronchoconstrict and dilate, sphincters in the gut, and all of that stuff. So it becomes a little tricky when we talk about smooth muscle. And a lot of mechanisms are on smooth muscle, and they're very similar to each other, to the heart too, except instead of using troponin C, smooth muscle uses the myosin light chain to control contraction. But the starting process is pretty much the same. It depends on calcium. And contraction, we'll find, is going to be smooth muscle contraction is going to be mediated through a G sub Q linked protein after activation of either an M3 or an alpha 1 receptor. So M3 being parasympathetic, alpha 1 being sympathetic. And G sub Q works a little bit differently. Just like before, or maybe we've seen this before, I don't remember. Active G sub Q will bind to a GTP, but G sub Q does not bind adenylate cyclase. It binds to phospholipase C, beta, or just phospholipase C. And PLC will cut the GTP into GDP just like adenylate cyclase did. But the most important thing is the effects on lipids. Phospholipid, so it's an enzyme that, phospholipase sounds like phospholipid. It's an enzyme that cuts phospholipids. They're going to cut phospholipids in the cell membrane to create two separate lipid compounds. The first one is a niacyl triphosphate, and the second one is diaglycerol. And diaglycerol actually stays in the cell membrane, and it works to activate an enzyme called protein kinase C. Well, protein kinase C phosphorylates things, as we saw that protein kinase A did. Remember, protein kinase A we saw in the G sub S pathway. Protein kinase C we see in the G sub Q pathway. But in this case, it can phosphorylate calcium channels to opening, causing an influx of calcium. Niacyl triphosphate works on the endoplasmic reticulum or the sarcoplasmic reticulum to release calcium from those calcium stores. Remember, these are smooth muscle cells we're talking about, so they're also going to have sarcoplasmic reticulums. Remember, in the G sub S pathway, voltage-gated calcium channels triggered the opening of the sarcoplasmic reticulum and the calcium influx. In this case, it's IP3, a niacyl triphosphate, that causes direct opening of these things. But the end result is the same, elevation of cytosolic calcium, which will rise and associate to calmodulin, activating myosin light chain kinase, which phosphorylates myosin light chain and allows the muscle to contract. But it gets a little bit interesting and even more complicated when we want to relax smooth muscle. Does anybody see a parasympathetic muscarinic receptor located on the smooth muscle cell membranes that cause relaxation? No. We do not tend to use these systems for that. Okay? And remember, cholinergic and muscarinic are the same thing. But let's see what happens. Well, binding of epinephrine or norepinephrine to beta 2 receptors or dopamine to dopamine D1 receptors activates a G sub S stimulatory protein linked receptor. Just like before, G sub S will activate adenylate cyclase, which will create an increase in the levels of cyclic AMP. Cyclic AMP goes over and activates a protein kinase. Now, don't mix this up with the heart. In the heart, we saw that the elevation of cyclic AMP promoted calcium increase from release of the sarcoplasmic reticulum, but also promoted calcium storage in the sarcoplasmic reticulum by driving that ATP calcium pump. And this is because to increase heart rate, we need a balance of calcium release and storage. But here, we find that protein kinase A will work to pump calcium out of the cell via ATP calcium pumps or into the sarcoplasmic reticulum, which will drive down the levels of calcium in the cell. And like I said earlier, we don't see any cholinergic or muscarinic receptors in the membrane that promote relaxation. That's because, for the most part, relaxation of smooth muscle is actually mediated by something different, known as nitric oxide. And from this point forward, you can think of nitric oxide as being the great dilator in the body, okay? No matter what it touches, it likes to dilate things, okay? Nitric oxide is an uncharged, small molecule which can easily and passively diffuse across lipid bilayers. And in smooth muscle cells, nitric oxide works on something very similar to adenylate cyclase. Okay, it's called guanylate cyclase. Guanylate cyclase. It works to increase guanylate cyclase activity, and instead of elevating cyclic AMP, it forms cyclic GMP. Which is cyclic guanine monophosphate. And for the most part, cyclic GMP has the exact same downstream effects as cyclic AMP. And elevated levels of cyclic AMP or GMP will decrease activity of myosin light chain kinase, decrease phosphorylation of myosin light chain, and cause smooth muscle relaxation. But where does nitric oxide come from? It actually comes from Nancy neurons, NANC, non-adrenergic, non-cholinergic neurons, okay? Recognize that the parasympathetic nervous system is involved in the relaxation of smooth muscle in some tissues, but the effect is not mediated through muscarinic receptors. So mediators like nitric oxide or vasoactive inhibitory peptide, VIP, are what mediate that smooth muscle relaxation. And these mediators are activated by the inhibitory parasympathetic neurons, those NANC neurons. A great example of this is the sphincters in the GI tract and the urinary tract, which relax and open via nitric oxide. Nitric oxide is also really important in the corpus cavernosa of the penis, causing vasodilation and the ability to get an erection in men. And now that we have talked about generic smooth muscle, let's take a look at some scenarios where the sympathetic nervous system and parasympathetic nervous system have specific smooth muscle actions, like blood vessels. Remember, blood vessels are surrounded by smooth muscle that can contract and decrease blood flow through the area, or relax and increase blood flow through the area depending on where the blood flow is needed. Interestingly, the parasympathetic nervous system actually does not have any direct innervation to blood vessels. But the receptors are still present, likely an evolutionary breadcrumb, like our appendix, for example. So the receptors, muscarinic receptors, are still there, but the innervation is not. And so imagine that this is a blood vessel. Okay, here's the blood, and we know our blood vessels consist of endothelial cells, which face the inside of the blood and have direct contact with our blood. And although the parasympathetic nervous system doesn't have direct innervation of smooth muscle cells, receptors are still present on both the endothelial cells, and we'll find them on the smooth muscle cells as well. Acetylcholine that is floating around in the bloodstream that hasn't been deactivated by that butyryl cholinesterase has the ability to bind to and activate M3 receptors here, which will lead to the elevation of calcium like we saw before. But vascular endothelial cells have a specific enzyme in them called nitric oxide synthetase, which can be activated by an elevation of calcium to produce nitric oxide from arginine. Remember, I said nitric oxide can easily and passively diffuse across lipid bilayers because it has a favorable partition coefficient. So it can easily enter those smooth muscle cells and activate guanylate cyclase, elevating cyclic GMP, decreasing calcium levels, and promoting blood vessel relaxation. It's important to note, again, that nitric oxide has a very short half-life and can only promote local effects. It cannot go out into the bloodstream and have effects in other places. And although this pathway seems a little bit more intricate between the two different cell types, the idea is pretty much the same as we saw before, right? But what happens, let's say somebody poked a needle through this blood vessel and this blood vessel got damaged, or the endothelial cell was scraped off by a needle or a white blood cell that was coming through. um, what would you expect in a case where something is damaged? You would expect the smooth muscle cell to contract. Anytime we damage anything in our body, we tend to contract it up to prevent blood loss, for example. You can see in this case that the role is to relax. But if that endothelial cell was scraped off, we also have M3 receptors on our smooth muscle cells. Now that smooth muscle cell has direct contact with the blood instead of being insulated by that endothelial cell. We saw before acetylcholine caused relaxation, but you can see these receptors are G sub Q linked, which increase calcium and cause contraction or vasoconstriction. Those same, it's also the same as well as the isotype being M3. So when it comes to the sympathetic nervous system, it does directly innervate blood vessels. And depending on where you're talking about in the body will depend on what receptors are located at those areas. Alpha-1 receptors are by far the most important, and those are located in most vessels in the body, especially the skin, mucus tracts, and kidneys. The goal with vasoconstriction is to increase blood flow to vital organs like the brain, lungs, heart, and muscle. Because if you think about it, when you're running away from the tiger, you want your blood flow to shunt to your brain, your lungs, your heart, and your muscles. Beta-2 receptors are found on smooth muscle in some arteries that go to muscle, which makes sense because we learned beta-2 a couple of slides ago. We learned that beta-2 receptors cause dilation, okay? So if it's in the lungs, it'll bronchodilate. So it makes sense to find beta-2 receptors in places like the lungs or the muscles, the arteries that go to muscles. And we saw beta-1 receptors are, they increase heart rate and contractility, but we also have some beta-1 receptors in our juxtaglomerular cells in the kidney, promoting renin release, which increases the renin-angiotensin-aldosterone system to increase blood pressure. And so all of these responses here are going to increase that fight or flight response. In the lungs, we have M3 receptors, which when agonized cause bronchoconstriction of the smooth muscle surrounding our bronchi. And we also have beta-2 receptors, which we saw promotes bronchodilation, like I said a couple of times now. So beta-2 activation in the lungs will cause bronchodilation. The eye poses an interesting area of study with the autonomic nervous system only because there are so many little darn automatic muscle functions that happen in the eye. For example, the iris circular muscle, for example, contraction is mediated by M3 receptors, which promotes miosis or pinpoint pupils. And contraction of the radial muscle is mediated by alpha-1 receptors, which induce mydriasis. Mydriasis. Remember, when running away from a tiger, you want to be able to see at night, so you want the dilated pupils. And the parasympathetic nervous system also promotes contraction of the ciliary muscle via M3 receptors, allowing for the accommodation for near vision. But the sympathetic nervous system the alpha-1, really only has a minor relaxing effect on the ciliary muscles. And the ability to use the autonomic receptors in the eye becomes important when we talk about clinical conditions such as glaucoma, which is going to come back over and over and over again. And if you don't know what glaucoma is, it's basically having too much pressure inside of the eyeball. Remember that aqueous humor is secreted by the ciliary body, and it flows through the anterior chamber, and it drains off via the trabecular meshwork and the canal of Schlemm, okay? 85% of it, so, right? And then there was another drainage pass as well through the uveoscleral route, which drains only about 5 to 15%. So, what things can increase pressure in the eye? Well, slow drainage. And glaucoma is often associated with an elevated intraocular pressure to the point that it starts damaging the optic nerve or the retina. Well, meiosis, if you think of meiosis induced by muscarinic agonist, that can actually stretch the iris and increase the rate of drainage, right? Via that uveoscleral route by opening up the angle, right? Opening the angle. But if you contract the ciliary muscle, well, this muscle is going to bunch up all right here, and it can actually potentially contribute and to the effects of glaucoma. The GI tract also has a lot of autonomic receptors. And one thing I want you to think about before reading over this is parasympathetic nervous system. What would you expect it to do to the gut? Well, it's rest and digest. So I would expect it to increase GI motility. And I would expect it to relax sphincters in order to promote defecation and things. And all of those are ran by muscarinic M3 receptors. Therefore, the sympathetic nervous system, I would imagine it doing the opposite. And that's exactly what happens. What we find is that alpha-1 receptors will contract smooth muscle and sphincters to decrease defecation, because you don't want to poop or pee while you're running away from a tiger, for example. Alpha-2 receptors and beta-2 receptors also promote smooth muscle relaxation, okay, which is going to decrease peristalsis. The urinary bladder is actually a point that's still kind of on the cutting edge of things. One thing I want you to think about is we have a lot of different muscles here. We have the detrusor muscle, which when it contracts, it squeezes on the bladder. And we also have the sphincters, okay? So if I was thinking, without looking here, parasympathetic nervous system, the parasympathetic nervous system wants me to pee. So in order to pee, I would have to contract the detrusor and relax the sphincter. So what we'll find is that M3 receptors are going to contract on this, the detrusor muscle, contract, and M3 receptors on the sphincter is going to promote relaxation via nitric oxide like we saw before. On the flip side, we would expect that the sympathetic nervous system does not want us to pee, right? When you're running away from a tiger, you don't want to pee. So we would expect the sympathetic nervous system is going to relax the detrusor and contract the sphincter. And it contracts by alpha-1, and it relaxes by beta-2 and beta-3. Beta-3, this is one of the few places in the body you'll actually find beta-3 receptors. When you think about the uterus, do you want to have a baby while you're running away from a tiger? No. So you'd expect that the sympathetic nervous system is going to have the relaxing receptors, which is that beta-2, like we saw in the lungs, like we saw in the bladder. And it's going to contract sphincters, versus the parasympathetic nervous system is going to have that M3 receptor promoting contractions. In the male genitourinary system, we have a couple of subtypes of receptors that are really important. We have the same things going on here. M3 receptors are going to promote detrusor muscle activity, as well as sphincter relaxation. But there is a specific area of subtypes of alpha-1 receptors that occur in the prostate specifically. A lot of times in men, they can get what's known as benign prostatic hyperplasia, where the prostate overgrows, okay? When it overgrows, it can cause contraction in here, which can cause obstruction of urine that cannot pass through. Well, I told you here that alpha-1 is the thing that causes contraction of smooth muscle. Here we're talking about ejaculation, but on the previous slide I said alpha-1 causes contraction of the sphincter. And the same thing happens here, except with the prostate, it's a specific subtype, alpha-1A receptors. You need to know that one, okay? Because what we'll find is we can use drugs like alpha-1A antagonists to antagonize the contraction here and promote urine flow in patients with this issue, okay? Another thing that's important is erection and ejaculation. So the parasympathetic nervous system is what promotes erection, and the sympathetic promotes ejaculation. One way you can remember this is point and shoot. So point for PNS, penis. Parasympathetic nervous system. Shoot for SNS. Okay, so the so the sympathetic nervous system is important for that, right? The 3 F's: fight, flight, and sexual intercourse. Also, the penis could be potentially something that somebody could grab a hold of in a fight, so you wouldn't want to have an erection while fighting, right? Anyway, another important place is the metabolic effects, and this is where I was pointing at the liver earlier. Anytime we have activation of the sympathetic nervous system, we're going to expect increased mobilization of energy storages, okay, from the liver especially, and that's all mediated by beta 2, okay? The last things we'll talk about is secretion. So if you think about it, secretion in the GI tract is going to be mediated by the rest and digest system, mainly M3. Bronchial secretions are also going to be parasympathetic nervous system, okay? Sweat glands, though, are the ones that are mediated by the sympathetic nervous system, which are mediated by acetylcholine muscarinic receptors. Remember the one place that's kind of weird. But those apocrine glands, the ones that stink, like in your armpits, are alpha-1 mediated, okay? One last thing I want to talk about before we end this lecture is it's important to recognize where those alpha-2 receptors and those muscarinic 2 receptors were located. Remember, these were the 2 receptors on the presynaptic sites where they essentially inhibit neurotransmitter release. We saw these 2 receptor subtypes being on the presynaptic, like adrenergic cell membrane, for example. This is where the alpha-2 was, to inhibit further noradrenergic receptor release, or in muscarinics, or cholinergic processes, this is where the M2 receptor was. And these 2 receptors work as that feedback inhibition of neurotransmitter release. And this is how it's done. So when norepinephrine binds to the alpha-2 receptor, or acetylcholine binds to that muscarinic 2 receptor, they're both linked to a G sub I inhibitory protein, which will decrease cyclic AMP production and decrease the action of protein kinase A. We know protein kinase A usually was the one that phosphorylated calcium channels and allowed them to open, causing increased levels of calcium. But with decreased pKa, calcium channels will stay closed. And both of these receptors are also, they also have direct signaling with potassium channels via their subunits, okay? We saw in the diabetes lectures how these potassium channels here remained open to increased membrane potential. And I just want to go back and show this again. We know that these stayed open to increased membrane potential. Well, activation of these receptors is also going to further hyperpolarize the cell and increase that membrane potential even further. Remember, it took depolarizing the cell in order to open these calcium channels when we were talking about diabetes, and there's no difference here. Therefore, both of these mechanisms is going to cancel out or block the opening of these calcium channels, leading to decreased levels of calcium, which will stop the exocytosis of those neurotransmitters of acetylcholine or norepinephrine, like we saw back here. That's where this is going on, okay? This here is a summary slide for you to start, getting all of these in mind on what they're doing, on the different G proteins and what they do, as well as whether M3 and alpha-1 generally increase cytosolic calcium, beta-1 and beta-2 generally increase cytosolic AMP, and M2 alpha-2 generally decrease cytosolic AMP, like we saw back on this slide. All right. Thank you very much.